Okay guys, so today we're going to talk about plant and animal cells. These are pictures that are in your notes of plant and animal cells, and you are responsible for knowing what these organelles look like. They kind of float around in this jelly, so they're not always in the same place, so I don't worry about the location in so much as kind of memorize sort of what they look like. And I'll be helping you with that along the way, but this is a great study tool, this first little set of pictures. All right, so we're going to start off with the cell membrane or the plasma membrane. It is selectively permeable. That means it only allows things in that it wants to allow in, and it basically keeps outside things outside and inside things inside. It has some glycoproteins and glycolipids for communication. It has some proteins in it. It has, the proteins are for binding and adhering, and there's cholesterol that hangs out kind of in this bilayer here. And we're going to talk a lot about the plasma membrane in the next one, so I'm not going to spend too much time on this now. But that's the cell membrane, which is on the outside of all animal cells and is just right inside the cell wall of a plant cell. The cytoskeleton is kind of like our skeleton. It's going to maintain the support and shape of the cell. It's going to hold all of these organelles in position, move organelles. It is involved in cytoplasmic streaming. It interacts with different extracellular structures. And we have microfilaments, intermediate filaments, and microtubules are the three different types. But I'm not going to ask that you know what the difference is between the three, just that they're part of the cytoskeleton. Now, this is the nucleus, this big thing right here. And it has little holes in it called nuclear pores. And inside, it's going to have these sort of this thread-like stuff called chromatin, and that's genetic information, that's DNA, that is uncoiled. So it's just kind of floating about, but it is bound in this membrane-bound nucleus, which is a phospholipid bilayer, just like the cell membrane. Okay, so it has that like an outer part and an inner part. It actually has two phospholipid bilayers instead of just one. All right, so nucleus, nucleolus, this is like where there's a high concentration of genetic information or RNA. And then chromatin, you got the thready DNA over here, nuclear pore, how things get in and out, and nuclear envelope, basically the boundary of the nucleus. The nucleus is usually the largest organelle. It is where you'll find DNA and DNA replication. It's also where DNA is transcribed into RNA, and therefore you have a nucleolus, which is where we begin the assembly of the RNA. And then I talked already about the chromatin, the threads, and when they condense down and they're going to start to look like X's, which we'll see in a few units, we call that chromosomes. All right, so an endomembrane system consists of an endoplasmic reticulum, a rough endoplasmic reticulum attached to a nucleus. You have a Golgi apparatus, and then you have these transport vesicles. So we're going to kind of walk our way through the endomembrane system. So first you have the endoplasmic reticulum, which is a network of interconnected membranes. You have a smooth ER and a rough ER. This rough ER has ribosomes, and ribosomes are where we have protein synthesis. So protein synthesis actually begins sort of on the outside of this endoplasmic reticulum with these ribosomes. Once the proteins make it inside the endoplasmic reticulum, they're going to be modified and tagged, and this makes so much more sense when we get to transcription and translation of DNA. But basically we're going to add a cap and some stuff to protect it, and then it's going to move to the Golgi apparatus. The smooth endoplasmic reticulum actually doesn't have anything to do with proteins because it doesn't have ribosomes on it. Okay, this is going to chemically modify small molecules, usually lipids, such as drugs and pesticides that come into your cells, This is the site of lipids and steroids. All right, the Golgi apparatus is right next to the endoplasmic reticulum. So the proteins are going to get tagged. They're going to get tagged by the rough endoplasmic reticulum, and it's going to be sent to the Golgi apparatus. And the Golgi apparatus is actually going to take in the protein, and the protein's going to go, if you'll follow my cursor right here, go all the way through the Golgi apparatus until it forms a lysosome or a vesicle. And it's basically like FedEx. It's going to package and sort and distribute all these different proteins. It adds little signatures like carbohydrates to the proteins. Now, lysosomes also originate from the Golgi apparatus, and they contain digestive enzymes. So this is where when a cell eats something, takes it in, these are going to, it's like the stomach. These lysosomes are like the stomach of the cell. They're just going to break everything down so that the cell can use it. And this is kind of what a Golgi apparatus looks like underneath an electron microscope. So much fancier microscope than we have here. You can see it looks like a stack of pancakes. And then you can see the vesicles kind of coming off of it. These are all vesicles that have sort of come off of the Golgi apparatus. So a macrophage is basically just a big, huge cell that eats things. And basically, they have a lot of lysosomes because they are going to take in things that they want to eat. We have a lot of macrophages in our body, and they protect us from the outside world. If a bacteria gets in, these cells will eat it like Pac-Man, pull it in, break it down, and spit it out. In eukaryotes, molecules are first broken down in the cytoplasm, and then they enter the mitochondria, where we are going to make ATP. And so cells that require a lot more energy, like your brain cells and your muscle cells, they have a lot more mitochondria than everywhere else. And the mitochondria has an inner and an outer membrane, much like the nuclear envelope. And you're gonna learn a lot more about the mitochondria when we get into taking glucose and converting it into ATP. So this is like a power station, like MLG&W or something like that. Gives us our power. And this is what that looks like under an electron microscope. So now I'm going to specifically talk about organelles found only in plant cells. So plant and algae cells contain plastids that can differentiate into organelles. So these plastids are typically like just sacs for storage. Now, the big deal is these chloroplasts, which look like stacks of pancakes with little lines coming off of the side. And this contains chlorophyll, which is a pigment and is a site of photosynthesis. And photosynthesis is where energy is converted from light energy to chemical energy. So we take the sun's light and we create food for the plant. The chloroplast has two membranes again. So we've seen the nuclear envelope that has two membranes. We've seen the mitochondria has two membranes. And now the chloroplast has two membranes. And this is just giving you an inside look at a chloroplast. Probably on the test, I will not ask you these questions, but you will be responsible for them in the next unit. So you might as well take a look at them. The inner, there is a stack of pancakes called granum. One individual pancake in this stack is called the thylakoid. And so you have thylakoids in a stack called grana, and then the stroma is like the cytoplasm of the chloroplast. Vacuoles actually, in plants have a lot more vacuoles. Animal cells tend to have vesicles, which usually contain like a digestive enzyme. Vacuoles are more just like a storage facility. So they put waste products in there, or they put some kind of chemical that if a animal bites into their leaf, that it just explodes out. It also stores water in there. And so there's a lot of different reasons why you would have a vacuole, but plant cells typically have vacuoles and animal cells typically have vesicles. So unicellular organisms that need to get around are probably going to need something like a cilia, which are these little hair-like projections, or a flagella, which is like a whip on the back. So these are some other items from a cell that you might be asked to know. A plant cell also has a cell wall. A cell wall is basically a thick protection. It provides support, but it also limits volume. So the cell can only take in a certain amount of water. And when it has enough water, those cell walls make it rigid so that the plant can stand up. But when it's dehydrated, that's why plants like slump over, because the cell walls are kind of caving in because of lack of water. It acts as a barrier to infection, and contributes to growth and development. All right, then outside the cell, there's a few things we have to know. The extracellular matrix that consists of a bunch of substances secreted by cells. It's basically going to hold cells together in tissues. So like our different tissues in our body, our muscle tissue, our skin, or whatever, those are all made of tissues, and tissues are made of cells. And so cells are all knitted together with this extracellular matrix. It filters out materials, and it keeps cells sort of in a similar location so they don't just float about. Cells also have communication devices, so you will be asked to know these 4 different types of cell junctions or communication devices. A tight junction is basically where they're just basically knitted together like a quilt, and this prevents substances from moving through any spaces within the cell. So this kind of prevents anything from moving back and forth. Desmosomes are going to hold cells together but allow things to move in, so it's a little bit further apart. Your gap junctions are channels that are going to run between membranes, so different cells can share information rather easily. And a plasmodesmata is specific to the movement of water, ions, small molecules, and it's essentially they use them a lot in plant cells because you'll have a cell wall, and so the plasmodesmata right here allows 2 plant cells to be connected to each other.